process of DNA replication.
Protein forms two hydrogen bonds and this is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make two new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase, because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make
So that's how DNA is replicated in bacteria. But fundamentally, it's the same as this alpha, beta, gamma, those that we talked about, 9 different classes. There's a special type of DNA polymerase which replicates the ends of the chromosomes, which we call them the telomeres. The telomeres are the ends of the chromosomes. This is replicated by telomerase enzyme. And you need to know that telomerase enzyme, the gene that codes for telomerase enzyme is switched off in almost all of the somatic cells, except those cells that have the capability of stem cells, or they are having the properties of stem cells, or stem cells like hematopoietic stem cells in bone marrow. So any organ, any tissue that has a stem cell in it, those stem cells have their telomerase gene expressed, i.e., their telomerase enzyme is working. So there is no shortening of the ends of the chromosomes. In other somatic cells, the rest of the somatic cells of the human body, telomerase gene is switched off. And that's why, one of the reasons why we get aged by the pass of time is because of telomere shortening. Every time a cell divides, or the somatic cells, I'm not talking about stem cells, that's a different issue. Every time a somatic cell divides, its ends, the ends of the chromosomes get shortened by a few bases. This is called telomere shortening. And this is one of the reasons why people get aged. Otherwise, if there were no shortening of chromosomes during cell division in somatic cells, people would have stayed at a certain age without getting aged. It's one of the theories of aging.
Research. We are now able to see how DNA is actually copied in the
Proteins replicate DNA in the process of DNA replication. Double strand by helicase. There is synthesis, continuous synthesis of one of the strands. It's called leading strands, leading recognition. So this is going through the whole process. The other strand is lagging, activated dual, the German, lagging behind. That one is replicated in a discontinuous manner. Every time the template strand for the lagging has to come towards the replication fork machinery, helicase, the DNA polymerase, etc. A short stretch of DNA synthesized called Okazaki fragment, and then it loops back again by a back stitching mechanism, coming forward and backwards. That's how the lagging strand is synthesized. And the only reason is because the lagging strand synthesis, which has to still be from 5 to 3' direction, is moving away from the replication fork. That's why it's synthesized in a discontinuous manner. Now, how DNA is packaged in the chromatin or in the chromosomes? We said 2 coils of DNA wrap around a protein called histone, and this histone protein is made of 8 subunits. The most important thing, because DNA and RNA have negative charge because of the phosphate in the backbone, they need a protein that has lots of positively charged amino acids, such as histidine, lysine, arginine. And it tells us that histone protein is rich in these amino acids, in these positively charged amino acids. Otherwise, it would have not been possible for DNA to wrap around histone protein.
او هناك او كولابوريتيف مشان يحس علاج
أو بكتيريا.
اولا التطوير كجات مثل برايمري
Primate DNA primate.
primary
Telomerase can only elongate from a free 3' hydroxyl.
موبایل بگم چه در باشه دنیا بیناموس بسازه. وقتی بیسی به موبایل بکاربینه چه در باشه. اتوانی امضاش کرد یا میکنه بسازه. بعد خدا که تیکلش ده اصلا. وانا زمان كبير بس ان شاء الله نسي قلت انا مشروع مكان او ركان قلت انا مشروع مكان. So we try to understand how genes are read and how much of that gene is read in human. So gene expression is not a simple process in human. It's not compared even with bacteria. Bacteria is much simpler. In human we have levels, different levels for regulation of gene expression. So we have two basic processes. The gene itself and how it's read to make an intermediate stage molecule, RNA, and then if that RNA is the mRNA type, messenger RNA, how it codes or translates the message into protein. So the genetic codon and translation process. So cells are simply able to read the genome through gene expression. All the 25,000 genes that we talked about we have on the DNA. Some of these genes are read, some others are switched off, maybe temporarily, maybe for life. That depends on the functional stage of the cell. So gene expression encodes either RNA and then stops there, or it makes an RNA as an intermediate stage to make protein and then stops there. All of these to dictate cell function, to tell cells what to do or what the functions are for the cell. Maybe it's an enzyme, maybe it's a hormone or a protein in a pathway, or it's an essential protein for synthesis of something else. So all of the functions of the cell are dictated by gene expression and how much of that gene is expressed in a cell. If if we define gene, it's nothing more than just a sequence, a primary sequence of DNA. But there are regions that we can distinguish from a DNA sequence as a gene or to be part of the gene. And there are other sequences that comes after and before the gene that are regulatory sequences for the gene. They regulate the function of the gene. So if you define a gene, it would be a nucleotide sequence in DNA that acts as a functional unit. So the gene itself has areas, but the whole gene altogether is a functional unit to make an observable product. And that observable product could be RNA, either catalytic, regulatory, or structural RNA, because we have over 20 different types of RNA, or it could be a protein. In the old books of molecular biology, this is how biologists define gene expression, is production of a protein. But that turns out to be wrong. The definition is missing something. We later knew that gene expression could produce RNA instead of proteins. And these RNA can be structural, such as the rRNA in ribosome, but they can be catalytic or carry a function, regulatory RNA, or carry a function, like messenger RNA, they carry a message. So gene expression is production of RNA or protein by a gene. So based on this fact, we have two types of genes, according to the function, what they do. They make protein or they make RNA. We have protein coding genes, probably slightly less than 25,000 genes are coding for proteins. So majority of our genes are coding for proteins. And there is approximately 3,000 of these genes among the whole genome are coding for RNA. We call them non-protein coding genes. Or in other words, you can call them RNA coding genes. Now we have another way of classifying genes. This is important because think about cells in different case scenarios, in physiological states or in disease states, pathological settings. Cells, whether they are healthy or diseased, they still need to carry out some basic cellular function and activities. So we have housekeeping genes. They keep the house tidy, means like, so housekeeping genes, constitutively expressed genes. These genes are expressed at all times. If you are awake, if you are asleep, if you are diseased, if you are healthy, if the cell is happy, if the cell has any pathology, etc. So these genes are constitutively expressed under different conditions. That's why we call them housekeeping genes. If by any reason these genes stop expression, cells simply die. Because if you have no housekeeping genes, then it means the house is going to be ruined and there will be chaos. And cells attempt suicide by apoptosis or other mechanisms. So it's needed for maintenance of basal cellular function. The other type is conditionally expressed genes, inducible, or genes expressed under regulatory conditions. So for example, a child, when they sleep before 8 o'clock, 8 p.m. I mean, their growth hormone release will be at the optimal. So that's needed for growth of bones, for growth of the organs, etc. So the timing that a special hormone is released from the brain, like growth hormone from the anterior pituitary gland in the brain, those are inducible genes. And there are certain circumstances, they are released under certain circumstances. Inducible, it's not always released. Adrenaline, you go to an exam, before the exam you feel irritable, you sweat a lot, you urinate frequently, your heart pump rapidly beats. All of those features are features of an anxiety. It's because of a hormone called adrenaline, a fight and flight hormone. Once you see the questions and start to settle down, and then you feel better. little by little. This is because the adrenaline is reabsorbed back to cells. So this is an inducible example of an inducible gene expression. Again, another example, when you take sugar, then insulin starts to get released when you have a high blood sugar in order to allow this sugar to be taken up by the body cells, insulin. So that's an inducible example of inducible. And the other genes that are expressed constitutively is housekeeping. We have something else called sense-antisense strand. Remember when we said the number of genes are actually much more to fit on a single-stranded molecule. That's one of the reasons why we have DNA as a double strand. So one strand may carry certain gene, and the opposite strand will still be carrying some other genes. So the strand which is carrying the gene is called sense strand. The opposite strand will be antisense or template strand. Template for what? It's template for the RNA molecule. So when you have gene on the top strand, like in this figure, so that is the gene itself. Sense strand. You can call it positive strand because it has the gene. But why, who knows why the opposite strand should be a template strand? Although this could be another gene, maybe another gene starts from here. So this gene starts from this point, maybe gene A to produce protein A. Another gene could start here to produce a different thing, a different protein or a hormone. Why this strand becomes template? I mean, it could have been the RNA could have been produced from the gene itself. Remember the complementarity, the base pairing rule? The RNA has to have the same sequence of the gene. So if this base is A, what you have opposite to A is thymine, T. And T opposite to T you have A. Opposite to G you have C. Opposite to C you have GG in the sense strand. So look at the RNA. As if the RNA has the same sequence as the gene itself. We need to have the exact mirror image of the sense strand in the RNA molecule. So this is the first step of gene expression, making an RNA molecule. The other gene is in opposite direction. There you have the non-template strand or the sense positive, the gene. Then the opposite strand becomes template. Template for the synthesis of RNA. Products of gene expression. Genes could produce RNA and then through this intermediate mRNA produce proteins. It's called central dogma of molecular biology. We go from DNA to RNA, RNA to mRNA, and then to protein. Product of gene expression could be still RNA, but without making protein, such as making telomeric RNA. Remember, the enzyme telomerase has an RNA with it. That RNA is still a product of gene expression. Ribosomal RNA, etc. These are structural RNA. That's not pekateva, shikl, pekata. They are not functional, but they make structural components like rRNA, ribosomal RNA in ribosome, telomeric RNA in telomerase. But these are catalytic or regulatory sequences of RNA. Messenger RNA is catalytic, it carries a message. Transfer RNA, it transfers amino acids in cytoplasm to the ribosome, the translation machinery. Small nuclear RNA, small interfering RNA, microRNA, kRNA, etc., etc. So slightly over 20 different types of RNA exist in the human cells. And most of RNA, we encounter RNA in the cytoplasm and sometimes in the nucleus even. Because we don't have a problem with the RNA, it's not like DNA. DNA one of the reasons why DNA is secluded in the nucleus is because it's liable for attack by water in the aqueous environment of the cytoplasm.
دكتور هو عفوا سلايد دكتور هسه هنيك يعني هو ولتي ار ان اي هنيك لاوانيك لان يعني اظن ان كودينج جينا كان اوانيك هو هر اكل البرداري كنا ولا كودينج جينا وات البرداري كنا ولا ترانسليشن كان بروتين بروزبل يعني اوش ماري كدان راو عفوا اوش ماري كدان راو ما بس اوي بو بيان جزائر ما بس اوي اماني جربيته او سياسه جربيته
من بروتين كودينج جينا كان معنا من بروتين كودينج جينا
داش خوش والله بس دكتور يسبونتي ار ان اي باش
لما من بروتين كودينج جينا نحمل
أزال نن كودينج والله هرب الخو هرب بجداري أكان ولا كودينج لا
بجداري
كواتب وش مارك بيش من جزارك أتواني بلين أو بيش من جزارة بهماشي وهندك إيش لكودينج كان قلت واتني
لا غلط مثلا protein coding genes are these genes it should make it must make a protein otherwise it's not a protein coding gene
دكتور بقول منا mRNA
mRNA carries the gene, the message.
That's good. Well, mRNA actually.
This is not an issue. Listen, this is not an issue of argument. It's a fixed fact. Okay, we call it fixed fact. راستيكا لا تنكا مجال مناقشة ثانية حتى راسيتين الوهية mRNA, tRNA, all of those, even rRNA, they participate in the process of translation, making protein. But is it itself a protein? Is tRNA a protein? Is a ribosomal RNA a protein? They participate, they help in the process of translation, but it's not a protein. Even mRNA is not a protein. tRNA carries the amino acid. If tRNA were a protein, then I would say it would have not been composed of A, G, C, U. It would have been composed of amino acids. So we have two basic processes of gene expression. Transcription is making an RNA from DNA, RNA molecule from DNA, any of those RNA. And the second process is translation, making protein from the message that's read through the genetic codon. Now there are steps between those processes. I highlighted only transcription and translation. These are the two major steps of gene expression. Going from transcription to translation through mRNA, we have something we call processing, RNA processing. RNA has a gene for translation, for protein synthesis. So the RNA, the mRNA that's made inside the nucleus during the process of transcription, that is immature, amylin. So we have to add a few things to it. Cells need to manipulate it. The hypoprotein will take you in a cell. The balancing the attack. When you need to so we need to prepare ourselves to prepare the mRNA. That's for RNA processing. It includes capping, includes splicing, it includes removal of introns, etc. So after protein synthesis, I don't know if people from protein expert people have said this to you. Some proteins need what we call post-translational protein modification. After translation, some proteins need the addition of a phosphate group or maybe something else, an additional chemical group. This is called post-translational protein modification or sugar or glycosylation. So all of these processes are happening after the process of translation. That's why we call post-translational modification of proteins. A protein can be some having a variable. شبيه خالصة of glycosylation and any given lipoprotein. شوفي مثلا يعني وشوفي phosphate مثلا. But these are the main process of gene expression, transcription and translation in DNA. You have preparation of RNA, the process of RNA processing, and after protein synthesis, not for all proteins, but some proteins need post-translational modification. originality. Why not all the 25,000 genes that we have, or let's say between 20 to 25,000 protein coding genes, are not expressed in every single cell of your body? Would it be possible for neurons, for example, to make antibodies? That's not possible because we know only big lymphocytes terminal mature to plasma cells, and these plasma cells start to produce antibodies and secrete these antibodies. So every cell in our body, as a somatic cell, has a specialization. خارجية جرازنا ولا خارجي مشاهينا كالأفراد يكون معرض شوية كالسكين خارجي اشتكوا أكاد هو Replication Fork. This is the other way. Excellent. So we have proteins. We have proteins, specific proteins bind areas in the gene called promoter. We'll come to that. These proteins determine within the transcription factor, this is specific transcription factors, the regulated transcription factor. Found in every classical protein, methylate transcription factor. It's process transcription. RNA, DNA. This is the first step of gene expression. Process again, transcription. Protein and transcription factors. The transcription factor. The general or basal, which is sort of little specific type of data. Specific transcription factors are the ones that determine which genes to be expressed in which cells of the body. Central phenomena is the HT. Now I'm trying to central manager or issue. Central manager. Approximately 208 different types of cells. So each cell will express a certain set of genes. Housekeeping genes now. Housekeeping genes is for all the cells of the body. Without exception. Remember we said if these genes are not expressed, then there will be no trace of cellular functions. Not on any individuals, not on any machine, not on any fluid, not on any metabolism, not on any virus given now. So the only thing that determines differential gene expression is the specific transcription factors within the family of transcription factors that bind genes or regulated sequences in genes. So this notion, we call it central dogma of molecular biology. So anything you need to know about DNA, RNA, and protein is around this scheme. It's oversimplified scheme to tell us how genes are read and how cells are reading these genes and how much of it is read. Genomic to describe the total genes we have on the 46 chromosomes, and that will include all of the chromosomal genes and mitochondrial DNA, RNA intermediate stage transcriptomics, around 3,000 maybe, more or less, and then the protein coding genes. If I were to go into the lab and study only proteins, I would be calling that area, that field, that specialty proteins. I'm going to get a paper with that and transcriptome on the side. That transcriptome is it's talking about the total RNA content of the cell, any type of RNA. Protein, any protein in the cell, if you study them, they have proteins. So cells with the genome expression in this scheme. What symmetry will do is like the angular structure in DNA pathogenesis, Watson and Francis H. the co-discoverers of the structure of DNA. Actually, they are the ones who put this scheme. They said we have the genes on DNA, carried on DNA, and these genes are through the process of gene expression, through transcription, and some of them through translation also. So they probably 5 or 6 years after the discovery of the structure of the DNA, they made this statement, and it turns out to be true. Now, how much DNA goes on proteins? See how much of DNA, how many sequences of DNA are in the favor of protection of the gene or regulating the gene. كسر 80 شخص صار 11 صار الجين very تشريف بس عندنا وإذا with all these harsh conditions in the environment, with all this stress around us, all these diseases, all these viral infections, cancer, exposure to toxic chemicals, to radiation, to sunlight, the stress of life itself, but you can still survive because your genes are still functioning. They want to survive. They want to perpetuate the species. Because our genes, the gut has created us, mighty gut has created us in a way that our genes have lots of levels of protection. One level is that DNA is double-stranded. Another level is DNA inside the nucleus secluded from cytoplasm even. Another level is it's packed around histone proteins in chromosomes. Unless you need these genes, then they won't be at want. Even if there is an error or mutation, there is a repair system for you. The gene itself will not be read and expressed unless there is a regulatory sequence that comes before it to tell the gene what to do and different other things. Talking about the typical eukaryotic gene, a gene itself will be made of two areas, transplant region or coding region have regulatory sequences or regulatory regions. This is a gene. One gene would have something we call a promoter. The start of the gene, a promoter, and then coding region and the termination sequence. It's nothing more than just a sequence of repetition of ATGCs. But how cells know that this is a gene? By knowing that this is promoter, that's coding region, and the termination sequence. This is been studied thoroughly in bacteria and human cells. Within this promoter, there is a specific repetition of few bases, few nucleotide bases, that would tell us the enzymes and the transcription factors, and this is the start of the gene, so the promoter. One of these areas in the promoter is called TA adapox. Okay. It comes before the coding region. So when the RNA polymerase, not DNA polymerase, RNA polymerase that polymerizes RNA polymerase scans the DNA. And when it comes to TA repetition, it would sense that as the beginning of the gene, the promoter. That's what we call promoters. Promoter is the area for binding of RNA polymerase and transcription factors. This is a little bit more emphasized. Promoter, how we use promoter, it has a sequence that allows upstream, upstream of DAT, we have something else called CAF box and GC box. It's minus 100, minus 80, minus 25, then minus 1, and then plus 1. There is no 0. Minus 1 is the end of the promoter. So from minus 100 to minus 1 is the promoter region. How we know that it's the promoter? How the enzyme knows this is the promoter? Because it has GC box, CAF box, and TATRH. How we know that? Regulatory sequences. This is promoter. RNA polymerase is the other step. Plus one change enzyme. It's the golden start of the coding region. Serotype and fully protein
What binds to the tau box? Is it the transcription factor or is it the RNA polymerase itself?
Both of them. Both of them.
Together they bind.
Yes. And the transcription factor from the transcription factor is only the general cause based on transcription factors. Because upstream of the minus 100 there is still a sequence of DNA upstream of the minus 100, which is called regulatory sequence. It has a name actually. It's called enhancer. And a protein called specific transcription factor called activator binds that enhancer. That protein activator, which binds the regulatory sequence enhancer, determines which genes to be expressed in which cells. For example, if I have hepatocytes, I classically get a protein albumin polysaccharide. The activator for albumin protein is found in hepatocytes, but the activator protein, which is specific transcription factor for antibody production, is not found in hepatocytes, it's not found in neurons. You get it?
The activators change based on the cell.
Yes, activators are specific transcription factors. Those are the ones that determine the differential gene expression. Within the coding region, we have areas called exons, areas called introns, exons. These introns, it's true that they are still within the coding region, but they are not coding for anything and coding for proteins. They are all non-coding sequences within the coding region. Exons are in contrary the coding regions within the coding sequences within the coding region. upstream, downstream, these terminologies. Anything comes before the gene goes upstream, after the gene downstream. The promoter region is the site of molecules, most transcription factors, basal transcription factors, and the RNA polymerase. This scheme is actually illustrates the trigger, the initiation, that's basically transcription. So we have transcription analogy and promoter educator, TNT box. And before the TNT box, you have CAAT and GC. Our petitioner, underlying, I think it was the lane, ATGC management. طبعا نفس الإجابة كمان لو جينا نعمل بالصبر نعمل أشتغل على التبيان تفتكر عندنا شوية This is under regulation. It's how the genes look at TNTA goes then I have CAAT and MGC. TNTA goes first thing RNA polymerase will be transcription factor. Transcription factor and to the RNA polymerase and to other proteins, other mediators. So if you pass on a specific transcription factor, the germs activated on the here enhances sequence. Bacteria vascular, aggravatum, chaniega, gene expression, void expression. What are mechanic neuron events? Antibody uska, initial, auch genetic antibody conversion, RNA polymerization, amino acid, transcription factor, and the cell over the parameter over the expression for this RNA. The specific transcription factor that activated is the one that determines and triggers the initiation of transcription. Here, the rest is super, there's Sodium, Rochium. Here, Here, شاء ما كان أصلاً تيجون في ذيك كيف برا لازم أيضاً تقول ناشر الشاكة ذيك اللي أدوانا وشيء دوانا This is a cafe. How we respect classrooms. This is discipline. It's not just about the result. That was the It's not understood. Activators are the ones that determine which genes to be expressed. All of this is what you see. The basal factors that are the polymerase find every gene in the world because they continually scan DNA and they can recognize and find DNA promoter regions. But not having the activator means there is no gene expression. Having the activator for a specific gene that tells us or tells the cell which genes to be expressed. But the protein and DNA protein there was by the way, like the sequence to the gene. But some sort of very good. I don't know what's going protein in the DNA, protein. So this thing is called transcription bubble, 5%. The DNA can't do it again, DNA can tear it down. Original replication. This is called transcription bubble. The bubble that gets run now transcription bubble. So RNA is built from small DNA. You have the ATP, DCTP, DGTP, DTTP. deoxy, the building blocks are deoxy, but here we have ribonucleotide triphosphates, A, U, G, and C. But sure, just this animation and then we can take a break. This event chain replication, the whole process is not separated into phases, into stages. جاء إقرار وتقبل بس بينما كان عمل الاختبار initiation and anticipated elongation termination. It's not like that in reality. It's all one go, one process. When it starts, it ends at the point. But for simplicity to study these processes, we have dissected them into initiation phase, elongation, and termination. And in each phase, there is something happening. All of them are happening very quickly. Initially, the cytoplasm somehow we need to hide it in the cytoplasm, or protect it in the cytoplasm. And this comes in two ways. From first ends, we need to cover the mRNA through the 5' end and something called target acid protein. It's a specialized type of guanosine called 5-methyl guanosine cap. The cap structure capping, it's called capping, the first process. And the 5-methyl guanosine G guanosine methyl acid. الرسالة معنى بدأت يكمل وزيادة بس هو شوية G. One of the G's and the 5' called 5-methyl guanosine. It's capping. That's how we protect the mRNA from attack on the 5' but about the 3' which involve the mRNA. The tail of the mRNA, which is the 3', there will be an addition or repetition of that A. That's why we call it poly-A tail. Poly-A tail, poly-attenuation. How many adenates in human genes? There are these possibilities between 20 to 200 adenates. And one is that subadenine beta gene heads adenine مثلا mRNA مثلا وليس from the 5' from the 3'. This is to protect mRNA. وإذا أردنا أن يسوي يسوي يكون cytoplasm divides splicing. Remember when we said the coding region has exon, intron, we have in the handouts. Exon, intron. The exons are the coding sequences within the coding region. These are the sequences which has the instruction to tell the translation machinery later to make the proteins. Introns are the non-coding regions. Introns are the non-coding regions. But why we have introns in the first place if they are non-coding regions? Who knows? Protein from قلنا كابوجيتي لغة splicing remove all the introns. Removing the introns. So yes, the introns are regulatory sequences within the gene, within the coding region of the gene. Remember, before the gene, upstream of the gene, downstream of the gene, we have regulatory sequences. Even within the gene itself, there are regulatory sequences. So one of the functions of introns, one of the functions of introns is they are regulatory sequences within the coding region of gene. This is one. Two, introns protect genes from the effect of ionizing radiation. يعني كبتدرش من تشكي UV X-ray gamma ray وهكذا أقول جينا كانت ينتهي مشكلة زي complementation. One of the things that protect genes from the effect of ionizing radiation is having introns. 2. 3. Introns help what we call alternative splicing. So introns allow genes to have spliced variants. يعني زي مش تصنيفين هذا ما يدري إكسون إنه زي ما كان زي إكسون ما كان بالإنترون أو بالإنترون هكذا ترى كذا في الرأس إكسون كان زي ما بخانة لو البروتين جاز بالمسكن شو يمسك كلنا جاء كم إكسون يكمل إكسون يمسك جاري كي أجمعنا إنسان يمسك بسنة كريم وتيجي جواز مسك أما صار كان هذا دشة في مكان one gene but eight protein products from one gene have one gene or three products from one gene so نزل يمسك one gene phospholipase 8 the thyroid gland and one gene هنا sequence DNA mRNA كشيء يعني sequence DNA بس advanced classified as classified يعني again intron لما تكون DNA كفاش بسنة introns having introns would not every human gene. Some of the genes have this capability making different proteins from one gene because of having introns. Introns allow the splicing allow فيخرج زاد يكون منوعين alternative splicing in certain human genes and the gene data. Because of having introns allow different combinations of exons to make different proteins. Splicing set of rules. Yes, I family, okay. I have the possibility that there are several versions here. لماذا كان يجب الأمر أشوار ليس كانت تكون أنتوان كان شرشة ياسع therefore the figure matter for the 20 standard amino acids that we have the 4 bases in DNA A, T, G, C 4 by 4 to the power of 3 that is just 4 possible codons 12 bases there A, T, G, C and 7 at least amino acids يعني شيء يعني ما يصير هذا الكودون يجي يبقى ياسع كشوة أو شيء يصير ما تقول أمامي أو كان for details مشروع this is after removal of neutrons code 1 genetic code 1 genetic code 2 etc. So every few letters it's telling to me one minus which minus is to specify. AUG is always standard in the beginning of every protein. This is called start codon. AUG specifies exactly. According to the rest of the genetic table, AAG goes for lysine, UUU goes for threonine, GGC for glycine, and we have 3 stop codons. UAA, UGA, and UCA. So these 3 stop codons are not coding for any amino acid. So if this is how every protein must have a start point and an end point. This is translation. It's making protein. So how do you read that genetic code of table? It's not something to remember from your heart. Just be back with the memory that would have been. So I'm going to have the argument and the end of this lecture. It starts with the standard and how it is. AUG always goes for defined. This is fixed fact in bacteria, human, and plant, and animal. Every creature you find it on earth. When they make proteins to the translation, the first codon must be must be defined, and the codon is AUG. That's for other reasons. AUG is the start codon goes for defining. The rest, at least you put it. I can tell you what I can it's stable. It's like calculating you have a complex mathematical equation you need to calculate. If I give you a sequence of DNA on mRNA and tell you to make a putative protein, then I have to provide you with a genetic codon table. So, أقل الإنترنت قال أنواع تيبل وأنا أدرك شو شعار وشرع شربات هم يكتب يكشف. So, أقل وقت ما نسمي فريق جسيم شو فريق بسيطة. سي رجلي وما يوم أوليزة سيناء C, U, C, A, C, G. So 4 codons, 4 possible codons making one amino acid, but only one codon is the methyl. For adenine, aspartic acid, glycine, arginine, glycine, threonine, leucine, isoleucine, arginine, and there are 3 stop codons. U, A, G, and then U, G, A. They make the 3 stop codons. So when the translation machinery encounters any of these 3 codons, mRNA gets understood how I was going to go. mRNA gets understood how I was going to go. I was going to go. سنة جملة أم جاء سنجلوتايد يعني الكودون سيدان الكودون كودون يكون خلال الدفاع جاء كودون سنجلوتايد جملة أو مسكت كودون كتير سنجلوتايد جملة كودون كتير أكاد يشتري بروسيس أو يتكلم عن لا dissociation يعني أو لا يبرز Characteristics of genetic codon. Genetic codon classification. They have some specificities. They are specific. So a specific codon always code for the same amino acid. CCU always codes for now. It's the same, but it's the same human. That's why it's called specific. Universal. It's the same in all organisms. Degeneracy. You have redundancy. يعني بدون زيادة. احنا بس نتشكل. وهذا ما يصير في mutation بشكل الكيمياء كان. مثلا mutation في بعض it's irreversible. Maybe someone gets a mutation in CUU, and the CUU changes to CUC. The genetic code has changed, but the amino acid hasn't changed. So that's, in that case, you can survive with that mutation without getting any disease, because still the same protein will be produced. Same normal protein, it's not a pathological protein. But imagine what if this CUU mutated to CCU1, this your acid gives C cytosine, so the amino acid changes from UC to protein. That's one of the benefits of having degeneracy. Degeneracy means redundancy, is to protect your genome, is to protect the protein coding genes, is to give you a chance to survive with lots of mutations. You can survive with lots of mutations without getting cancer, without getting any diseases. This is one of the reasons. I have more than one genetic codon for one amino acid. That possibility gives you a chance to survive better than harsh environments. Non-overlapping and commas. There is no comma, there is no punctuation. It's not overlapping. And except that I go codon you get the same. So between one equals the next one, there is no punctuation, space line, comma, not overlapping, not necessarily. Lecture guide, photo, not not overlapping, and commas. This is the RNA. type of the RNA which brings amino acids to the mRNA in the translation machinery. Has some modified basis and basically mRNA translation machinery ribosomes. But with ribosomes except on what endoplasmic reticulum, ribosomes are not seen assembled like that in cytoplasm. It has subunits, small subunits, large subunits. They are separated into three floors in cytoplasm. Once there is an mRNA, then they get assembled. Initially, the mRNA uses GTP hydrolysis to attract the small ribosomal subunit. And then the large subunit joins the complex. And this is ready for translation. We have three regions: E for exit, P for polypeptidyl, A for amino acid. Now ribosomal protein لما كان الأعضاء الأمريكية يعني سيدنا UPA مفهومة فرضية ولا بس الأساسي شيء الأساسي process the ribosome moves into the right hand side, bringing the mRNA from 5' to 3' direction. So the mRNA 5' to 3', the whole ribosome moves every time from left to right, from 5' to 3'. How many nucleotides, 3 nucleotides are triggered at a time? E is empty initially, P is empty, A takes the first amino acid, then moves to B side, and then the next tRNA comes carrying this next amino acid, depending on the genetic code. And then this amino acid is given to the already existing amino acid in the P side. Writes for B from polypeptide because it starts to make the polypeptide. So it's teaching the amino acids together through what? Through peptide conformation. 5 and each-time-fold peptide translates. The P side amino acid معناها ستيارة شعار كيريا شعار amino acid تنشتر من الأيوة أو amino acid كيريا ستيارة كيريا amino acid بيت ونسين كيريا كيريا تنسين كيريا كيريا translates. أو شيء ونسين peptide conformation. Just appears tRNA into the draw. So the P side exit. العمل يكون يسجد الأمانة السنة في البشرة. الشجاع يشكل نفسه لازم مقابل الأمانة السنة. In the tRNA, there's a region called antibody that actually sits opposite, exactly opposite the equivalent. However, because mRNA equivalent, tRNA control nasio or nasio. There is a difference between ribosome in human versus bacteria. The reason why I brought up this slide, though we are not interested in bacteria, as medical students or as doctors, we're not interested in bacteria unless they are causing diseases. We are interested in human cells, to know more about human cells. But knowing about ribosomes in bacteria and just an idea about the molecular size would allow us to understand the mechanism of action of some antibiotics. Some antibiotics we take for infections affect bacteria by affecting the ribosomes but not affecting human because human ribosomes have a slightly larger molecular weight ribosomes compared to bacteria. Humans have 80 S. S is standard. It's not molecular but normal and molality. It's molecular size by S. I'm recognized that the ribosome in one capital centrifuge molecular size S one table standard. La bacteria الإنسان سيبا بنجاك الإنسان أصبح بشر حاجات أشكال هي سبب الجاكات حتى سبب الجاكات حتى شكل شخص الإنسان كذا أشكال واحد so flip 40s for small so if you have plus 60s that will be 80 and 30s plus 50 that will be 70s okay this is standard there is nothing wrong about that كل الشخص اللي قاعد يعمل غير أول شيء بيتراوح أجل الركوز ومقبل أن حسن الفيشيا حتى يسمع كشاني كشي كثير هذا كشي كثير ركوز الإنسان يشكي حتى يسمع but a simple ribosome large and small together if they separate the subunits they will have slightly larger size other mathematically it should be because 58 is supposed to be 100. It's not like that. Alone, separate subunits have different sizes, but the sample organism is different. Here's an ribosome that you use for RNA, RNA, ribosomal RNA, ribosomal RNA. أو يتجسد جزء آخر البروتين ويشكل أولا بعد كده نبقى على البروتين وعنده الميتوكوندريا الأسوسوم وفي البلازما التي جزء lipids membranes Let's go to the last thing. Slide again. Translation, the same like transcription. Listen, this is the brief. We need functional proteins, initiation factors, then the last subunit joins. Then you have the assembly, and then start translation by moving to nucleotides at the time in the 5'3' direction. This initiation, elongation, is about addition of amino acids to the drawing chain. This cycle, every time an amino acid comes in, makes a polypeptide on the drawing chain. We need hydrolysis of GTP. Once again, the DNA replication, there is a release of energy. The translation, we need energy. There is GTP hydrolysis and GTP phosphate is not always there. But the replication one here for أما الإنزيمات يعني لا تصنع فوقها عوضها أما رسمي العالمين وشو أما رسمي كورتيزول أما رسمي مشكلة العالمين شو أما رسمي الترتيب موجود جانب سيارة vertical transference. This is elongation. Then there is termination. Termination here termination achieved. So stop for the after. During termination, the stop codon is a place where there is no tRNA matching the stop codon. Something else comes in, which is called here's an release factor. يعني شيء يستوقف لنا كتيارين يعني مثلا تيارين التي يمكن أن تجعل ما نسيت
This guy again is still equivalent to tRNA and protein given release factor. هذا بكتيريا سيدانا معناها release factor 1 بزيد للخارج سهلة release factor equal eukaryotic release factor. Yeah, the process of translation, then translation. You understand that? That's the gene expression. And the last movie, again, the trailer in the translation.
Amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the large green molecules also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the glowing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E